Hello friends, now we are going to discuss the normal bone metabolism. The normal bone metabolism is a continuous balanced process by which bone is formed, resorbed and remodeled to maintain strength, mineral homeostasis and repair any micro damage. The following are included in the normal bone metabolism. The bone cells involved are osteoblasts, osteocytes, osteoclasts, bone remodeling cycle, and regulation of bone metabolism by hormonal control. These hormones include parathyroid hormone, vitamin D, calcium phosphate, and calcitonin estrogen. Local factors, that is rank-rankle OPG system. Rank is receptor activator nuclear kappa beta ligand and OPG is osteoprotegrin and the mechanical stress. Further, we shall see the differentiation of mesenchymal stem cells. The undifferentiated mesenchymal stem cells are converted to osteoblasts, cartilage, and fibrous tissue under certain conditions. Under conditions of low strain and high oxygen tension, the mesenchymal stem cells are converted to osteoblasts, and under intermediate strain and low oxygen tension, the mesenchymal stem cells are converted to cartilage tissue, and under high strain it is converted to fibrous tissue. And in this diagram here it is depicted the differentiation of stem cell into particular tissue. The key factors involved in conversion of mesenchymal stem cells are: for myocyte it is myo-D, and for adipocyte it is PPAR gamma, and for chondroblast or chondrocyte it is SOX9, for osteoblast or osteocyte RUNX2. The BMP, bone morphogenic protein, and WNT stimulate the conversion of stem cell into osteoblast or osteocyte. And in this image, the osteocytes are seen and the osteoblasts are also seen. Here it is the cartilage tissue showing the chondroblasts, perichondrium, lacunae, chondrocytes, and extracellular matrix. And the other conversion is hematopoietic progenitor cells differentiating into myeloid and lymphoid lineage. Myeloid lineage forms erythrocytes, platelets, neutrophils, eosinophils, basophils, monocytes and macrophage system. And lymphoid lineage forms B lymphocytes, which are converted to plasma cells, and T lymphocytes and NK cells, that is natural killer cells. Now we shall discuss about the osteoblast, osteocyte and osteoclast. These are the cells involved in the normal bone metabolism. The osteoblasts are derived from undifferentiated mesenchymal stem cells. The stimulating factors include RUNX2, BMP, beta-catenin, core binding factor alpha 1. The WNT stimulate osteoblasts. TNF alpha inhibit osteoblasts. Therefore, high WNT leads to high bone mass, whereas low WNT leads to osteopenia. Now, sclerostin and DKK1 Dickoff related protein 1 inhibit the WNT. Therefore, the inhibition of sclerostin and DKK1 increase the bone mass. Osteoblasts produce ALP, osteocalcin, type 1 collagen, bone sialoprotein, RANKL. These are used as indicators of osteoblastic activity. The increased ALP indicate increased osteoblastic activity in conditions like rickets, osteomalacia and healing fractures. The normal or decreased ALP indicate defective bone formation, example hypophosphatasia. We shall discuss about the osteocytes. Osteocytes are derived from osteoblasts entrapped in mineralized tissue. This is the osteocyte. These are the cellular process of osteocyte, and these are the cytoplasmic process projecting through the canaliculi. Osteocyte is stimulated by calcitonin and inhibited by parathyroid hormone. Calcitonin. Calcitonin inhibit the osteoclast and therefore block the bone resorption of calcium and therefore finally decrease the serum calcium levels. Whereas the PTH stimulate the bone resorption and increase the serum calcium levels. The osteocytes release sclerostin, that is SCL, and it provides the negative feedback on osteoblast mediated bone deposition. This is the image showing the bone metabolism. This is the osteocyte. This is the surface osteoblast. These are the activated osteoblasts, and these are the osteoprogenitor cells, and this is the osteoclast precursor. This is the activated osteoclast, and now the negative feedback mechanism provided by the sclerostin release from the osteocyte is shown here. The osteocyte release the sclerostin that bind to LRP56 receptor on osteoblast and inhibit the conversion of osteoprogenitor cells into active osteoblasts by blocking the WNT and beta catenin pathway for the formation of active osteoblasts. This is the flowchart depicting the image. Osteocytes release the sclerostin, which is a glycoprotein, and the sclerostin bind to LRP56 receptors on osteoblasts and inhibit the conversion of osteoprogenitor cells into active osteoblasts by blocking the WNT and beta catenin pathway, which leads to decreased bone formation finally. Now, the conditions like mechanical loading, hormones like PTH and estrogen decrease the sclerostin. It leads to decreased sclerostin levels, which causes decreased inhibition of WNT beta catenin pathway, and it leads to increased differentiation of osteoprogenitor cells into osteoblasts and increased bone formation. And in certain conditions like immobilization, aging, and steroid usage, there will be increased sclerostin. It leads to increased levels of sclerostin, and therefore there will be further increased inhibition of WNT beta catenin pathway, which leads to decreased differentiation of osteoprogenitor cells into osteoblasts, and it finally leads to decreased bone formation. Loss of sclerostin function, that is sclerosteosis, is associated with high bone mass because there will be decreased inhibition of WNT beta catenin pathway. Romosozumab, monoclonal antibody against sclerostin, it increases the bone formation and decreases the bone resorption, therefore used in osteoporosis. We shall discuss about the osteoclasts. These are derived from hematopoietic stem cells in monocyte macrophage lineage. The function of osteoclast is bone resorption. Now some basic points are: rank receptor is present on osteoclast precursor, rank ligand is present on osteoblast, and osteoprotectin OPG prevents the binding of rank and rank L. The bone resorption. This is the osteoblast with rank ligand, and this is the osteoclast precursor with rank receptor, and binding of the rank ligand and rank receptor leads to conversion of osteoclast precursor into activated osteoclast This process is termed as osteoclastogenesis and it leads to further bone resorption. This is the activated osteoclast, and here we can see the ruffled border of the osteoclast and lysosomes releasing the lysosomal enzymes and carbonic anhydrase and release of hydrogen ions. First of all, the osteoclast binds to the bone via integrins. These are the alpha V, beta 3 or vitronectin receptors. The bone resorption occurs in depressions, that those are the Haushi plaqueninae, synthesis of TRAP, tartarate resistant acid phosphatase, and release of hydrogen ions and lower the pH and increase the solubility of hydroxyapatite crystals, and lysosomal enzyme cathepsin K digest the organic matrix. The osteoblasts and tumor cells express the rank ligand. Osteoclastogenesis is inhibited by OPG, that is osteoprotegrin. Now we can see in the image depicted, this is the osteoblast with rank ligand released, and this is the osteoclast precursor with rank receptor, and this is the OPG, that is osteoprotegrin. Now here we can see that the OPG competitively binds to the rankel and prevents the binding of rank with rankel. Therefore, there will be inhibition of activation of osteoclast, that is process of osteoclastogenesis, and it leads to decreased bone resorption. The bone resorption occurs in conditions like multiple myeloma and metastatic bone disease. In multiple myeloma, the myeloma cells have increased rankel expression and decreased OPG production. Therefore, it leads to high rankel and OPG ratio. Therefore, there is increased osteoclast formation and thereby increased bone resorption. Therefore, in multiple myeloma, there will be presentation of lytic bone lesions, bone pains, pathological fractures, hypercalcemia. In metastatic bone disease, the that is the tumorous conditions like breast cancer, prostate cancer, renal cell carcinoma, there will be increased rankel expression by osteoblasts and decreased OPG levels. It leads to increased osteoclastogenesis and increased bone resorption. Denosumab is a monoclonal antibody against rankel. It prevents the binding of rank with rankel and therefore used in multiple myeloma, bone metastasis, and osteoporosis. Vicious cycle of tumors. Tumor cells have increased expression of rankel, therefore there will be increased osteoclast activation. It leads to bone resorption, and in bone resorption there will be release of growth factors. Those are TGF beta, IGF, and these growth factors stimulate tumor growth, and further the tumor growth causes increased expression of rankel and further osteoclast activation. This is the vicious cycle of tumors. Thank you.